Greetings to everyone. My name is Ubong David Ofion. I'm going to present the paper with the title Impacts of Photo Treatments on Tomato Physiological Qualities During Storage and On-Shelf Display. Tomato is a vegetable that is produced and consumed globally due to its health benefits and economic importance. Its well-defined developmental stages, genome, and enriched genetic resources makes tomato to be classified as a model for studying the behavior of fruits and vegetables. Due to its short shelf life, focus has been intensified on proper preservation method that will extend its shelf life and improve its quality. Photosynthetic active radiation is a portion of electromagnetic spectrum that is essential for photosynthesis. Its wavelength ranges from 400 to 760 nanometers. Plants absorb this light for their growth and development. While this light spectrum has been applied to plants artificially at the growing stage, its application to crops at post-harvest phase is limited. This spectrum of light has the potential of lengthening the shelf life, delaying senescence, and improving the quality of fruit and vegetables after harvest. They can influence the activities of enzymes, protein molecules that regulate gene expression, and can also control the transcriptional processes by activating or repressing their activities. The figure here shows how different spectrum of PAR light can influence the pigment of tomatoes. On the left, the blue spectrum, when exposed to tomato, excites a special protein known as the flavin adenine dinucleotide chromophore. This protein promotes photoexcitation of electron in tomatoes. This results in the activation of the cryptochrome receptor. The cryptochrome molecule undergo conformational changes and interacts with the transcriptional factors, in this case, the NOR-NAC and NAC092. The interaction energizes the transcriptional factors to suppress ethylene biosynthesis by directly repressing the expression of genes involved in ethylene biosynthesis pathway, resulting in reducing ethylene production and delaying pigment changes. On the right, red spectrum, when radiated on tomatoes, energize the SIH-Y5 transcriptional factor to activate the phytochrome receptor. The phytochrome receptor is responsible for changes in pigments from green to yellow, yellow to pink, pink to light red, and light red to red color through the production of different carotenoids. The activation of the phytochrome receptor stimulates the gene for ethylene biosynthesis pathway, which lead to increase in ethylene production, resulting in increased accumulation of carotenoid, hence accelerating pigment changes. The specific objective of this study was to investigate the effect of different spectrum of photosynthetic active radiation on the physiological qualities of tomatoes. And the quality parameters are the pigments, the firmness, the pH, and the total soluble solid. A few studies have been conducted on this subject. Wong et al. 2018 provide a theoretical background that photosynthetic active radiation can suppress the activities of peptide galacturonate, the primary enzyme responsible for cell wall degradation. Lorente 2016 identified red spectrum as a stimulator of carotenoid pigment. Lee et al. 2021 submit that photosynthetic active radiation can stimulate the transcriptional factor for sugar biosynthesis, leading to the expression of its gene and increase in sugar production. For the material collection, Sedang 2 cultivar of tomato was sourced from tomato growers and subjected to 3 light treatments: the blue, the red, and the white spectrum, at a temperature of 5 degrees Celsius and relative humidity of 88%, as used by Martinez Zamzora 2023. In an attempt to replicate the market condition, the temperature and relative humidity of various fruit and vegetable retail shops were measured, and an average of 22 degrees Celsius and 78% relative humidity was obtained. This condition was replicated in a climate chamber at the Control Environment Lab in the Department of Biological and Agricultural Engineering, University Putra, Malaysia. The samples were transferred to the chamber after 5 days of treatment, and the developmental progress monitored until it reaches the stage 5 of development, which is the light gray stage, as prescribed by the USDA classification. Here are the various parameters that were measured and the equipment used in its measurement. The result: for the measurement of pigment, the L, A, and B parameters were measured, where the L represents the brightness of tomato. From the figure, it could be seen that the red and the blue spectrum samples perform better in brightness than the white spectrum after treatment and during shelf display. The A value, which indicates the progression of color changes from green to red, reveals that during shelf display, the red and white spectrum accumulated more red color than the control and the blue spectrum. This aligned the submission of Lorente 2016, which their study revealed that red spectrum enhances ripening of tomatoes. But this study has provided additional information or findings showing that white spectrum can also enhance ripening in tomatoes, while the blue spectrum delays ripening. The B value represents the accumulation of yellow pigments, and from the figure presented, blue spectrum samples accumulated more of yellow pigments. Yellow pigment in tomatoes indicate the increase in accumulation of beta and the zeta carotene. For the firmness, after treatment and during shelf display, the white and blue spectrum samples were firmer than the red spectrum and the control samples. In terms of the pH is seen as a good indicator of quality assessment in tomatoes. The lower the pH of tomatoes as tomato ripens, the more resistance for tomato to resist microbial attack. From data presented during shelf display, blue spectrum samples had the lowest pH value, indicating that blue spectrum is capable of offering tomato high resistance to microbial infestation. For total soluble solids, which measure the sugar concentration in tomatoes, after treatment and at shelf display, the red spectrum samples accumulated more sugar than other spectrum. In conclusion, red and white spectrum hasten the ripening of tomatoes during storage and on shelf display. Blue spectrum delays ripening in tomatoes. Rather, it facilitates the increase in yellow and pale yellow color in tomatoes. Blue spectrum also enhances the firmness of tomatoes. It also reduces the pH value, making tomatoes more acidic and offering tomato high resistance to microbial infestation, while the red spectrum enhances sugar accumulation in tomatoes. In recommendation, I recommend that more study be carried out on other crops, and also an intense study should be carried out on the nutritional and phytochemical content of tomato to ascertain the impact of each of this spectrum on the nutrients of tomato. Thank you.